Mhm.
All right. Hello, everyone. Welcome to the Society for the Study of Reproduction webinar entitled Marsupial Reproduction, Lessons from the Pooch with Andrew Pask. My name is Ronald Fan, and I am the webinar specialist for SSR. Also with us is Jane Fenelon, who is your moderator for today. If you have any questions or concerns about this webinar, please feel free to reach out to me directly at SSR headquarters via email at info@ssr.org. Just a couple of housekeeping items before we get started. All participants are muted with the exception of the speaker and myself. The webinar is scheduled for 60 minutes and is being recorded. It will be posted to the SSR website shortly thereafter for members to access at their convenience. In just a few minutes, the moderator will begin with an introduction followed by the speaker presentation. Please feel free to submit your questions during the presentation using the Q&A text box on the bottom middle of your screen, and the moderator will read them to the speaker when the question portion of the webinar begins. And with that, Andrew, please test your microphone by saying a few words, and if a couple of participants, if they could let us know in the Q&A chat box that they can hear them loud and clear, we would appreciate that.
Hi everybody, it is great to be here, and hopefully I can tell you some interesting things about marsupial reproduction today. So hopefully you can hear me nice and clearly. Yep, that looks good. Thanks everyone.
Thank you everyone for letting us know the volume level is good. All right, over to you, Jane.
Great, thanks very much. I'll just share my screen. Okay, thanks everyone for coming along today. My name's Jane Fenelon and I'm a research fellow at the University of Melbourne. I'm currently working on the reproductive biology of the echidna, and I'd just like to take this opportunity to give a bit of shout out to the monotremes, as they're one species which we're not talking about in this webinar species. But I mention this also because our speaker for today, Andrew from the University of Melbourne, has also worked with another monotreme, the platypus, in addition to all the other species shown on this slide. Despite being born in the UK, Andrew has spent most of his life in Australia and his career to date working with marsupials. His first foray into the world of marsupials was for his honours and PhD at La Trobe University in Melbourne, Australia, under the supervision of Professor Jenny Graves, working on sex chromosomes in the Tama wallaby. For his first postdoc, he moved to the University of Melbourne, where he continued to work on the Tama wallaby with Professor Marilyn Renfrey, who many of you would know. In 2001, he was awarded a C.J. Martin Fellowship, which is a prestigious 4-year Australian medical fellowship that enabled him to spend 2 years working with Professor Richard Beringer at the MD Anderson Cancer Centre in Texas, before returning to the University of Melbourne. In 2008, he relocated to the US after being offered the position of Associate Professor at the University of Connecticut. And after 8 years in the US, he was enticed to return back to the University of Melbourne, where he's been ever since. And in 2018 was promoted to full professor. Slides aren't progressing. There we go. I first met Andy back in my honours year many years ago, when we were both in Marilyn's lab working on the Tama wallaby. The Tama wallaby is a great marsupial model that both Andy and I have worked extensively on, and I'm sure you'll hear more about that in the talk to follow. But I thought I'd just take this opportunity to explain a little bit about this particularly exotic species and some of the difficulties we have in actually working with an exotic species like the Tama. One of the first problems is you first have to find your wallabies. You can't order them online or from Jack's Labs. The University of Melbourne is located in Victoria and in Melbourne, which is around about here. And where we get the wallabies from is Kangaroo Island, which is this tiny little dot off the coast of South Australia, which is about an 8-hour drive to get to the coast, and then another 1-hour ferry trip to get to the island. Once on the island, we have our specialised 4-wheel drive and our Walla van, complete with wallaby. This van can hold about 100 wallabies, which we then transport back to Melbourne, or we can also use it as a bit of a mobile lab. And it's converted quite nicely, actually, set up to have at least three of us sitting in there at one time. Although it's quite hard work and you do work a lot of nights, there is certain advantages to working on Kangaroo Island too, as there's a fair bit of sampling of some of the local wildlife that's available. Of course, once you actually catch your wallabies and bring them back to Melbourne, it does make life a little bit easier. But then, of course, you do have to then go and get your wallaby from the colony whenever you want to do anything with it. And this is not what anything like what you would normally see for a mouse house. Wallaby yards are quite large and the wallabies are free range in the entire time. So here's just a little primer on how to actually catch your wallaby. First of all, you find a very large butterfly net, which you have to shoot out and catch a wallaby as it's running past you. Once you have caught your wallaby, it then needs to be transferred into a sack, which Andrew is demonstrating very nicely here. Though admittedly, he's looking like he might get kicked in the face by the feet at this point in time. And then once you have your wallaby safely in the sack, you are then able to check its pouch and see if it has any pouch young. In addition to his wallaby catching skills, over the years, Andrew has also won a number of awards, including from the Australian Society for Reproductive Biology and awards for research excellence and supervision. Since establishing his laboratory, he's received research grants from the NIH, as well as the Medical and Basic Research Councils of Australia, and has published over 120 research papers. He supervised multiple students, PhDs, masters to completion, and has featured extensively in the media. In addition to the Tama wallaby, he has on occasion been known to branch out into research on the mouse and even human. However, in more recent years, his main research has focused on work with a number of other marsupials, which you will hear about today. Over to you, Andy.
Thanks for that, Jane. There's some really old pictures of the yards there. That's amazing.
Yes.
I think Jane and I figured out yesterday that we've known each other now for 19 years. Is that right? Yeah.
Something like that. Too many.
It's been a long time. Okay, so this, let me just make sure I've got the right screen up. This should be right. Can you see my full slide there, Jane?
Yes.
Okay, perfect. All right. Well, thank you very much for that introduction. And yeah, I think, you know, something that all of us can really appreciate who work on exotic animals is just how challenging it is to just get to that point of actually having your animal to work on. We have to go through all of these things, as Jane had just explained, just to get to actually working on the animal species that you want to work on. It's so different from just walking down to the mouse house, ordering, you know, whatever mice that you need, and then just picking them up and doing the stuff in your lab. But I think there are enormous rewards that you get from working on exotic species, which is what I hopefully can highlight for you today, particularly for our amazing marsupial mammals. So what I'm going to go through today is some of the really fascinating and unique aspects of their biology, but particularly focusing on all of our favorite topic of their reproduction. So really trying to understand more about their own reproduction, but how our studies into marsupial reproduction have really helped us to understand more about our own eutherian mammal reproduction and also the evolution of sex determination across all vertebrates, and even why marsupials have such a strange mechanism of reproduction with this development of a young in the pouch. So I'm going to go through a number of different studies that I've done over the course of my career with marsupials, and then really talking about each of these really fascinating aspects of their biology. So the first question people often ask is, why would you study marsupials? So I think there are a number of things that are really fantastic about marsupials. Firstly, they're mammals, which is really nice in terms of comparative biology. So they're same as us. So in terms of understanding mammalian sex determination, you've got a nice mammalian model. But what's really unique about them is that they're very genetically distinct from us, or from placental mammals or eutherian mammals. They form a separate lineage of mammals that is really quite different. And that's fantastic for comparative biology because it gives us sort of an evolutionary step away from ourselves, which we can do comparisons with to start to understand some of the really highly conserved and fundamental mechanisms that are critical for driving mammalian reproduction and development. They're also developmentally unique. They have a really specialized mode of reproduction, and the way in which they develop is really quite different. And again, that affords a number of really amazing advantages for things that you can do in marsupials that are really difficult or impossible to do in other mammal models, such as mouse or rats or any other sort of placental or eutherian mammal. So just to explain how marsupials sit phylogenetically with the rest of the vertebrates, you can see in this phylogeny here, I might just get my pointer on. Let's go, laser pointer, here we go. So you can see here shown in red are the mammals. And mammals are formed into 3 separate groups. So you have the monotremes, which is what Jane was speaking about before, which are platypus and echidna. Then we have the marsupial mammals, of which there are a wide variety of species. And then we have our placental or eutherian mammals, which is shown out here, which is mice, humans, dogs, cats, everything else you can think of in this group here. So you can see marsupials are this really genetically distinct lineage of mammals, and they last shared a common ancestor with us around 150 million years ago. So it is really quite a deep divergence between us and marsupial mammals. So this gives us a really nice evolutionary distance to look across when we're looking for conserved mechanisms that are driving different developmental events. Monotremes diverged around 170 million years ago, so they're slightly deeper again at the branching of mammals. And these don't have internal gestation. So these lay eggs, and the eggs hatch to give birth to new little mammals. But in our marsupials and eutherians, these developed internal gestation. They both have a placenta, despite the fact that eutherians are also called placental mammals. Marsupials actually also have a placenta, but it's a much more simple structure. And I'll talk about why that's been so important in marsupial reproduction a little bit later in the talk today. And if we look at things that are similar between our eutherian mammals and marsupials, they both have an XY sex determining mechanism. They both have the SRY gene on their Y chromosome, so they appear to determine sex in the same way. It's upregulated right before testicular differentiation in marsupials, just as we see in eutherian mammals. They both give birth to live young, so they're viviparous. And as I mentioned before, they are in fact both placental mammals in that they both actually have a placenta that is really important for getting nutrition from mother to the developing fetus before birth in both of those lineages. Obviously, the one thing we always think about with marsupials about what makes them so unique is their really unique mode of reproduction. So it's characterized by having typically a very short gestation period and a very simple placenta, as I mentioned before, and this picture here shows that simple placenta. So this is it right before term birth, and you can see it's only 2 cell layers thick up here and 3 cell layers thick down the bottom here. So it's incredibly simple compared to a eutherian placenta. But then after this short gestation, they give birth to highly underdeveloped young, or our tritial young, which you can see shown here, that then at this incredibly early developmental stage have to be able to crawl into the mother's pouch. So they have to have really significant upper body development so that their arms can move and they can actually crawl to get into that pouch. And then they have to be able to attach to the mother's teat and start suckling straight away. So they have to have very good development of the head, the jaw, and the musculature so they can attach to that teat and start drinking mother's milk. And then they have a prolonged period of lactation in the pouch where they complete the majority of their development. So that's where the majority of their organogenesis and development and differentiation is really occurring, is during that period in the pouch. And they're really sort of like a mid-gestation mouse fetus, very early human fetus type stage at the point at which they're born, and then have to make that transition to developing external to the mother and in that pouch environment. But it's exactly this unique mode of reproduction and this our tritial birth, or this birth of the very small underdeveloped young, which makes them so amenable to all sorts of different manipulations that you just can't do in a eutherian or placental mammal. So some of the work that we've done has been to then treat these young with hormones from birth. And you can see here's an example of that shown on the right-hand side over here, where here's a newborn tammar wallaby in the pouch. And you can see we can take hormones, treatments, inhibitors, all sorts of things, and just deliver it directly to the developing baby. This is just in a little capillary tube. you just slide it alongside the mother's nipple, which is shown here. And as the baby's suckling, it will suck down whatever it is, the treatment that you're giving it, and it will give a very, you know, defined dose directly to that developing fetus, even though it's a baby, at that particular stage of development. This is much, much better and a much, you know, more finessed way of trying to treat and see what these things do to different aspects of development than you can do in a mouse. Because when you inject chemicals into the mum, you never really know what's making its way across the placenta, what dose is getting to the fetus, and how that then is impacting the fetus. Whereas when we treat a tammar wallaby in this way, or any marsupial, we can give a very prescribed dose that we know is going directly to the fetus during a particular stage of development, and then see how that's impacted the developmental outcomes. So in doing this, we've used various different treatments over the years to manipulate aspects of sexual differentiation, and that has really expanded our understanding of that particular process and how it works. And in particular, as I'll talk about today, the influence of hormones on that process. But you can also use this same process to manipulate aspects of all different parts of growth and differentiation, which, you know, many people have done, including my own lab over the years. And as I said, it's provided lots of insights, not only into how marsupials develop, but also more broadly into these mechanisms and how they're conserved across all vertebrates, and particularly our own sex determination, and how susceptible our gonads are to the influence of hormones as well. So one of the first questions I really wanted to ask in marsupials was to try and understand their mechanism of sex determination, and particularly the evolution of sex determining mechanisms across vertebrates. So if we look again, this is a simplified vertebrate phylogeny. You can really divide vertebrates into two distinct classes, which is the mammals and then everything else, all the non-mammalian vertebrates. In terms of how they actually determine sex, there's a very clear divide between those two groups. So in all non-mammalian vertebrates, they have a primarily hormonal influence that determines sex. So in fish, amphibians, birds and reptiles, which you can see all shown at the top here, they have a range of triggers or sex determination mechanisms, which range from environmental to genetic sex determining mechanisms. They have all sorts of different chromosomes and all sorts of different triggers in the ways in which they actually make those cell fate decision switches from being either male or female. But the underpinning of all of those mechanisms is it primarily works through the production of aromatase and then the production of estrogen in the very early embryo. So sex is really determined in all non-mammalian vertebrates by them either having the presence of estrogen, and then that actively drives ovarian development, or they don't produce estrogen and then you go down the male developmental pathway. Now, this whole mechanism has been completely flipped then when we switch to mammals. All of a sudden, mammals have only genetic sex determination. So we have genetic sex determination for all mammalian species across those three lineages. And there doesn't appear to be any role for hormones in this process. It seems to be driven only by the presence of some factor on the Y chromosome. For marsupials and eutherians, this is the SRY gene. It's still yet to be determined for monotremes. These guys are actually quite crazy with their sex chromosomes. They have 5 X chromosomes and 5 Y chromosomes, and it's still unclear exactly how sex is determined in these guys. But at least for marsupials and eutherians, we have that SRY gene as being the primary driver. And as I said, there doesn't appear to be any influence of hormones on that early differentiation process. So it's often thought that mammals are perhaps resistant to the effects of early hormones during sexual differentiation. So if we look at the role of estrogens in sex determination in mammals, so what we know is, just summarizing from that previous slide, is that estrogen is absolutely required to drive ovarian formation in all those non-mammalian vertebrates that I mentioned before. But studies in mice where they knocked out estrogen receptor and aromatase, which is the enzyme that actually makes estrogen, so these animals are all either deficient in estrogen signaling or deficient in the production of estrogen, they still were able to develop ovaries. Even though those ovaries failed at some point later in development, they were able to go through that initial phase of actually developing an early ovary. So we conclude in mammals that it was not required for ovarian formation, but it is critical for maintenance of the ovary. You will not get long-term survival of an ovary, and in fact, it will sex reverse into a testis if you don't have that estrogen signaling. But that early specification occurred in the absence of estrogen signaling. Interestingly, this sort of assumption or this dogma that there was no role for estrogen in early development was really sort of generalized to be something that we would, be expected to see for all eutherian mammals, just based on this study in mice. But in fact, if we look in humans and other animals, such as the goat that you can see here, it does appear that there is estrogen present in the very early ovary, and it might play a much more critical role more broadly in mammals, and it's really mice that might be much more resistant or not needed the effect of estrogen for early ovarian development. So they might be quite different there in terms of the way that they do things. But nevertheless, the mammalian dogma was that there was no real effect of hormones on early sex determination. And the reason that that was assumed to be the case is because, of course, what you're having in a placental mammal is you have to go through those early stages of sexual development in utero. So while you're inside your mother's uterus. And marsupials, just like placental mammals, when the female becomes pregnant, you do see increases in circulating estrogen levels. So obviously, if your gonad was incredibly susceptible to estrogen, that might impact the ability of a gestating fetus to then determine sex correctly. If you're incredibly susceptible to estrogen and you're going to have an influence of maternal estrogen, that's going to prevent you from undergoing normal male development. And so it was thought that perhaps mammals had evolved this sort of resistance or alternate mechanism to having an estrogen-based sex determination system so that they could undergo sexual development in utero. And that's what really enabled us to have that prolonged gestation and change the way in which we reproduce. So one of the things we thought would be really great with marsupials is because they give birth to such small young that we can treat and then observe the effects, is we can really test the effects of estrogen on very early development by directly administering it to young in the pouch and seeing then what happens to their sexual development. And in fact, this had been done a long time ago with researchers in America. So this was done originally by Burns using a model called the Virginia opossum, which I'm sure you're all familiar with, the ugly opossum. And this particular opossum, they were able to, when the babies were born, they just painted the back of each of those little babies in the mum's pouch with estrogen. And they were able to show that they could completely sex reverse XY individuals to developing as a female and developing an ovary. And this experiment was repeated some years later by Marilyn Renfree and Douglas Coveney in 2001, right when I was joining Marilyn Renfree's group as an early postdoc. And what they showed was exactly as Burns saw in the opossum, is that you can completely sex reverse Tamar-Wallaby gonads when you expose those pouch to estrogen, just with that little straw alongside the nipple, as I showed you in the picture before. And this is the histology of an ovary from a normal female, and you can see the cortex and medulla here. And then this is an XY individual, so an animal that should have developed testis that was exposed to estrogen. And you can see instead of forming testicular cords, we see a beautiful cortex and medulla. So you see complete sex reversal of the tissue and really very normal ovarian formation, just driven by exposure to estrogen in a mammal. It's not just the somatic tissue that sex reverses, but you can also see that the germ cells sex reverse. So they enter meiotic arrest in those estrogen sex reversed testes. So not only does the tissue form into an ovarian structure, but the germ cells also follow that female developmental pathway as well. So you're getting sex and soma both being completely sex reversed. So this is a true example of sort of a functional sex reversal, if you like, where you're actually getting that whole tissue forming a functioning ovary, even though it's an XY tissue. And this is purely driven by the presence of estrogen. So somehow this estrogen has the ability to then override that genetic sex determining mechanism being driven by the SRY gene to then, instead of sending that gonad down the testicular pathway, to completely reverse it and send it down the ovarian pathway instead. So this was something I was really interested in digging into deeper, was really understanding the molecular mechanism of how this is working. What does estrogen do? How is it causing this particular sex reversal to occur? And can that shed light on how estrogen is determining sex in non-mammalian vertebrates and the evolution, how we transitioned from this estrogen-dependent sex-determining mechanism in non-mammalian vertebrates to our SRY-dependent mechanism that we see in mammals. So one of the first things I did was look at the genes that are involved in male sex determination and female development and see what happens when we expose the gonad to estrogen. So as expected, we saw a suppression of male factors and an upregulation of female factors that are critical for granulosa cell development and other factors of female reproduction. What was really interesting though, was that we didn't see a change in the expression of SOX9 in the sex-reversed testis. Now, this was incredibly confusing to me when I first saw it. So SOX9 is the gene that is absolutely critical for driving testicular development in all vertebrates. It is the critical testis determining factor. All SRY really does is serve to upregulate SOX9, and then it is SOX9 that drives the rest of testicular differentiation. So when I was looking at the impacts of estrogen and how that's affected this developmental process, of all the things I expected to see suppressed by estrogen, SOX9 was top of my list. So when I saw that the messenger RNA, so at the expression level, this was not suppressed, I really struggled to understand exactly how sex reversal was occurring in these species. So what I did then was look immunohistochemically at what was happening with the SOX9 protein. And I found that although SOX9 was really abundant in the estrogen-treated gonads, which you can see on this bottom panel down here, as opposed to the normal testis at the top, you see SOX9, the difference was that this SOX9 was no longer in the nucleus of the Sertoli cells, but was rather relegated to the cytoplasm. So it was excluded from the nucleus. So this was really interesting because SOX9 is a transcription factor. It's required to upregulate the testicular differentiation pathway. And if it's not allowed into the nucleus of the Sertoli cells, then it can't have that role. So it can't activate male development rather. And so in the absence of activating male development, you go down that female developmental pathway. So this was the first time we started to understand the mechanism of how estrogen might be working, that was really excluding SOX9 from entering the nucleus in those somatic cells. So when we published this work and I started talking about it at various conferences, people were like, well, this is a weird marsupial thing, and that's because they're an ancestral lineage of mammals, and maybe they still have, some of the old mechanisms working that we see on non-mammalian vertebrates, but this isn't something that you would see in other mammals or placental mammals. So we then sought to investigate that. So I thought, okay, let's see, does this same mechanism work in a mouse? Again, as I was saying before, it's really difficult to assess in mice because if you treat female mice with estrogen, the placenta is actually really good at removing estrogen from the circulation and making sure none of it gets across the placenta to the developing fetus. So we couldn't really test it in that way. So the only way we could do this was by developing a gonad culture method. And this was done by Deirdre Matisky, who's a very talented postdoc in my lab, who's able to dissect mouse embryonic gonads at embryonic day 10.75, which is extremely difficult. And then we can culture them for 48 hours and we get this really nice development of the mesonephros and the gonad that you can see here after 48 hours in culture. You can see here's just a section through the cultured gonad and staining for apoptosis markers. You can see we don't see much cell death at all. So these are really nice, healthy gonads and mesos growing after 48 hours. And then we can stain for SOX9. So this is just a control culture and we get nice nuclear SOX9 in the forming testis cords, just as you would expect. So now we can take this system and we can directly expose the gonad to estrogen and we can see what happens in this mouse model. So you can see here again, here's control gonad shown at the top and all our SOX9 is nicely nuclear in those developing Sertoli cells as you would expect. If we expose to very low levels of estrogen, so these are sort of environmentally relevant levels that you might see from an endocrine disruptor in the water source or something like that, you can see that although most Sertoli cells have nuclear SOX9, we do see a fraction of them that have completely cytoplasmic SOX9. So some Sertoli cells appear to be more impacted than others, but when they are impacted, it's sort of an all or none response where you throw all your SOX9 then out into the cytoplasm. So in the absence of nuclear SOX9, this particular cell can't go down that male or Sertoli cell fate pathway and is more likely to be confused and perhaps go down that granulosa cell pathway. And you can see as we increase estrogen concentration, so these levels here are quite high, we see almost entirely cytoplasmic SOX9. We no longer see nuclear SOX9, and we can induce a much more profound sex reversal event in those particular gonads. And again, we looked at male markers which were suppressed, and then this graph down the bottom here is showing female markers which we saw upregulated after those 48 hours of culture, even with that very, very low level of estrogen. So it's definitely having an impact on the gonad. So it is telling us that although estrogen is not required in the mouse for granulosa cell differentiation or to trigger any sex determination event, if you have exogenous estrogen, if you're exposed to estrogen, it can still drive those in different gonad cells down a female developmental pathway, or at least confuse some of those cells in the gonad. After showing it in mice, we also wanted to see if this was also the case in humans. And so we switched to doing similar experiments, but now using NT2D1 cells. And these are a human testicular-derived cell line, a testicular teratoma cell line. And what we did with these particular cells is, again, just grow them in culture, expose them to estrogen. And we saw exactly the same phenomenon, that you saw that SOX9, instead of being nuclear, was rather now excluded out into the cytoplasm. And again, we looked at male markers and female markers and what was happening to them within these particular cells. And we saw that after about 48 hours, a suppression of almost all markers of Sertoli cell differentiation, and then an upregulation of female markers, just like we saw in mice and just like we saw in the wallaby. So now what we're looking at is an incredibly conserved mechanism where estrogen is actually able to affect somatic cell fate in the mammalian gonad and actually push it towards a female developmental profile. I really wanted to understand this mechanism further. And so, to cut a very long story short, I had a PhD student, Melanie Stewart, who was really looking at the fundamentals of the mechanism of how this works. And it turns out that SOX9 and SRY actually are initially trapped in the cytoplasm. And the way they're kept out in the cytoplasm is they're bound to microtubules. And then when they activate either sex determination or testicular differentiation, they become released from the microtubules, and then they enter the nucleus and then activate their downstream target genes. What we saw was that all of these cells, whether it was a human, a mouse, or the wallaby gonads, have a very significant and very rapid reaction to exposure to estrogen. Within just 30 minutes, those cells rapidly stabilize the microtubules, and more SOX9 becomes bound to that microtubule network. And so you can see here that when we treat with estrogen, this is a co-IP. So we've precipitated out tubulin, and then we've probed for SOX9. You can see a huge increase in the amount of SOX9 bound to the microtubules that we see here after we've exposed those cells to estrogen. We also see a massive increase in just the total amount of microtubules in those cells just after 30 minutes with estrogen. And we've been able to show now through the work of Mel's thesis that this is really all mediated through non-genomic estrogen receptor signaling and the MAPK pathway to rapidly stabilize that microtubule network and to really quickly sequester SOX9 out in the cytoplasm. So it's a very rapid mechanism that the cell uses to say, no, we're going to stop SOX9 from going in, so we're going to stop that male developmental process. And then after a prolonged period of time, you start to see the impacts of genomic estrogen receptor signaling, where that activated estrogen receptor then translocates to the nucleus and then starts to activate downstream female target genes. So it's sort of a double sort of approach where you quickly keep SOX9 out in the cytoplasm and then give time for that estrogen receptor activation network for the genomic responses to occur to start to then shift those cells towards that female developmental pathway. So when we come back now to looking at the evolution of vertebrate sex determination, we can see that although mammals have this SRY primary sex determining mechanism that's very actively driving testicular development, all of those mechanisms that are there in non-mammalian vertebrates for estrogen driving granulosa cell development are still in our genome. They still sit there, they're just not being activated. And so it's really SRY has come in on top of that and supplanted those mechanisms. You now no longer need to make estrogen in the very early ovary to drive its development. It can happen passively, and you're actively driving testicular development. But it remains that if you then have an estrogenic influence, it's going to push those cells down that pathway. So in revisiting this, we know now that we are not resistant to estrogen in mammals, but rather that's not the case. And that always causes some degree of sex reversal. And then this has very broad implications now for particularly endocrine disrupting chemicals, of which a very large number mimic estrogen, and certainly some of the most pervasive ones that we see in our environment, such as BPA and a number of different herbicides and pesticides, are all estrogen mimicking chemicals. And these are very readily able to cross the placenta. And so it raises a lot of concerns about what are the impacts of these chemicals on reproduction and reproductive health, not only in humans, but also in wildlife. So across the full spectrum of mammals, how are these chemicals now impacting their growth and development? So this was something then we wanted to branch out to and investigate in marsupials. One of the things that is synonymous with marsupials is farming areas. So they're incredibly abundant in farming areas. You can see just a couple of pictures down there. Here's a kangaroo in canola crops and in wheat fields, and they're a huge pest for the farmers here in Australia. And often they have big culls of the kangaroos in those areas to help reduce their numbers because they can completely you know, destroy some crops. But the problem with them being abundant in agricultural areas is obviously that's where there's a lot of herbicide spraying going on. There's also incredibly limited water resources in Australia. You know, we're a country that is stricken by drought very, very frequently. And so what that means is on the farms, they have dams and water reserves that are, you know, real popular water holes for all of the marsupials in the surrounding areas to come to. Often they're the only permanent water source in a very large area. And these accumulate large amounts of runoff and pesticides because we just don't have enough rain to really flush these chemicals away all the time. So when we do have rain, they accumulate in these very limited pools, and then they concentrate as evaporation removes all the water from those ponds, and those water resources get smaller and smaller. Another problem with endocrine disrupting chemicals is that they accumulate in fat, and particularly in breast tissue. And because the marsupials undergo a prolonged period of their development while suckling from the teat, they have the ability for those animals that are, you know, during their life consuming crops that have been sprayed with these pesticides, drinking from contaminated water resources. They're building up these endocrine contaminants in their fat stores, and then these travel across into the milk and might be impacting marsupial development. So that was something we really wanted to have a look at, is what are the potential effects of endocrine disruptors on marsupials. So this was the first study that anybody had ever done looking at a relevant endocrine disrupting chemical and impact on marsupials. And the chemical we chose to look at was atrazine. So this is a herbicide that's been banned in the European Union since 2004. In Australia, we use about 3,000 tonnes a year on Australian summer crops still. And in the US, it's about 20 times that amount that you use on your crops. So incredibly high amounts of atrazine are being used. In fact, I think in the US, it was the second most common herbicide used in the last decade. So a lot of atrazine is being used. Atrazine is highly problematic because it's very persistent in the environment, has a really long half-life in both soil and water. And importantly, obviously for our study, is it's known to affect reproduction and to have a feminizing effect on all vertebrates that have been examined that have been exposed to atrazine. So for our study, we took a group of tammar wallabies, and as Jane showed before, this is no easy feat. You know, you have to wrangle your 50 tammar wallabies that we had in this study and catch them all the time and treat them with the particular chemical that you're treating them with. In this particular case, we just gave them atrazine in their drinking water. So we just filled up the troughs in each one of those little fields with either atrazine or just regular water, our control group. And then we measured various aspects of their masculinization throughout development. Now, tammar wallabies take a long time to become sexually mature. They take about 2 years in a male. And so we followed them for the first 200 days of life in their pouch. And I'm showing you here a graph where we measured their penis length. And you can see atrazine shown in red, control animals shown in blue, that the animals that were exposed to atrazine had significantly shorter penises. This is a proxy measure of how much testosterone they're getting, what their masculinization is like. So it shows that we've probably had an impact on the production of male hormones, which is leading to that reduced length of the penis. And this was really very significantly shorter. Unfortunately, because of the developmental time of the tammar, we weren't able to follow this study on longer, but that's something we want to do with some of our new marsupial models. So as I said, this was the first time that anybody looked at endocrine disruption in marsupials, but we're able to show that atrazine, yes, indeed impacted male development, just as you see in every other vertebrate. So there's no real surprise there, but it was important to show that this does actually happen in marsupials, as well as in non-mammalian vertebrates as well. But these, you know, can have devastating impacts for some of our endangered marsupial groups, particularly because they are really often found in these agricultural areas. And we have lots of critically endangered marsupial populations, as well as heavily fragmented populations as well. And this could really tip the balance for some of them if they're not being able to masculinize normally during their life because of exposure to some of these chemicals. But as I said before, what we really need to do now is study the long-term impacts of these endocrine disruptors in terms of what's the reproductive output of those animals, what does spermatogenesis look like, is there ongoing health concerns for those animals? And so to do that, we really can't do it in the tammar wallaby, and we need to develop alternative marsupial mammals that we can use for this particular sorts of studies. And so the tammar, as I mentioned before, has been our primary model, which is this little wallaby you can see up here on the right. It has amazing genetic resources. It's a large animal, which has a lot of advantages. So it's quite easy to work on and to get big tissues from, and you can bleed them and do hormone assays and stuff because they are quite a large animal. But that also makes them very difficult to work on. I still bear the scars on my arms of being kicked by and bitten by many a wallaby over my life. They're very expensive to keep. So you can see from that facility that Jane showed pictures of at the beginning, you know, we've got them in a really huge outdoor grass, you know, many hectare facility that requires heaps of maintenance for the water and the fences and everything else. So they're very, very, and you have a full-time staff member out there looking after them. It's a very expensive endeavor to house and to look after Tama wallabies. The biggest problem for developmental biology studies, the kind of work that I really like to focus on, is that they're monovular, so they only have one baby. So if we really want to have a look at fecundity, you know, how this is impacting their fertility, they're only going to have a few babies over their life. So unless you're looking at the impacts of a particular exposure like atrazine on hundreds of tamas, you're never going to get significant data to be able to, you know, actually see what those impacts are. And that would be an incredibly expensive experiment. So it just makes it really difficult. Also, this long time to sexual maturation. So 2 years to become sexually mature in a male, 1.5 years in a female, is an incredibly long time to follow up these studies. Certainly not, you know, able to be done within the course of a PhD or something like that. So it makes it really, really difficult to do that. So other models that have been used in the past is the monodelphis. This is really quite common in the USA. But we don't really have them in Australia. We used to have a colony, but it was no longer supported. So we were looking for alternative models. And the one that my lab had settled on is using the Dunnart. And this is the Dunnart that you can see here. We work on this guy, the fat-tailed Dunnart. They're mouse-sized, which makes them very easy to work with. They have one of the shortest developmental times and the smallest babies. This is actually the palm of my hand shown over here on the right. And that is a newborn Dunnart on the day of birth. So they are absolutely tiny, incredible. They have multiple babies, so you can see down the bottom here, that's opening up the pouch. You can see they have lots of little babies. They're really easy to keep. They eat cat food, which is really nice. We're developing genetic and cell resources, which I'll talk about, and they then give us the potential to do developmental biology studies in this because they have so many similar properties to a mouse. They're really easy to breed in captivity, which is great. As I said before, they have a really short gestation. It's actually the shortest in any mammal. So it's just 13.5 days until they give birth. And then as a result, they give birth to some of the tiniest young of any mammal. And they've developed over the years, there's been a number of labs that have worked on them, and they've developed methods that you can actually culture that embryo right throughout every period of its development, right up until birth, which is really an amazing feat for a mammal as well. And obviously opens up a whole host of abilities then to go in and manipulate those young. Just to give you some perspective of the size of these things, this is like the little one I was holding on my hand before, and then this is what a mouse pup size is at birth. So they really are incredibly tiny. Now that has advantages because we can do all sorts of things to shift developmental trajectories of tissues, but obviously they're very difficult to work with because you're basically working on, say, an embryonic day 11.5 mouse embryo is about the stage that they are at birth. So they're quite fragile, quite difficult to do a lot of manipulations with. And across marsupials, you know, they fall into this group of dasyurid marsupials that give birth to the tiniest young. And these really are about 20 to 30 milligrams at birth. And this is in contrast even to Tama wallaby, which gives birth to a baby that's about 350 to 500 milligrams. They're almost mouse pup size at birth for the Tama wallaby, and they're much more developmentally progressed at birth than you see for the Dunnart. So this is going back another stage in development again, at which these animals are externalized and we can start to do developmental manipulations. This is just a quick snapshot of their colony. So you can see here, we just keep them in rat boxes. So very simple husbandry. Here's some of our breeding cages. So you can see the babies up here through a little glass window, but they're very easy to keep. They don't require any specialized husbandry or any specialized food really. You know, you have to watch a little bit of their nutrition, but they're very easy to keep. And in a room this size, we can have the same reproductive output of several hectares covered with wallabies, for example. So it's a really good way to do this. We're developing a lot of resources for this because we really want them to be a great model that people can use. So we're doing a developmental staging series, which you can see here. And again, just to show you, the mouse neonate is about equivalent to about a month after birth for the Dunnart in terms of its size. So really, really very, very different in size. We're also looking at the development of the whole embryo. So we've done not just that staging series, but we're also looking at skeletal development. One of the fascinating features of marsupials is they really have accelerated development of the anterior portion and delayed posterior development, as you can see here. This is an Alcium blue cartilage stain. So you can see they have cartilage in the anterior portion, but none in the posterior, and absolutely no bone on the day of birth. And they develop bone just 1 day later. And you can see that's mostly associated with the jaws and the arms that they need to paddle and to be suckling for milk as well. So they're amazing models, again, to study craniofacial patterning, limb development, and particular heterochrony, because they have this developmental timing difference. So they're fantastic models for developmental biologists. And just to expand their utility, you know, we've also done full micro CT scans of every stage of development that people can download and look at 3D models to really start to understand how they actually develop across all stages and how their skeleton develops. One of the first things I wanted to look at is when do the gonads develop. The Tama wallaby is about equivalent to a pouch young at about day 9 for the Dunnart. So this is when the Tama wallaby is born, is about this stage. And then they develop their gonads just a couple of days later in this punk stage, we like to call it here, where they get a mohawk. Somewhere between this stage here is when the Tama wallaby would develop gonads. So I was assuming that with the Dunnart, they wouldn't develop gonads until at least a week or 2 after birth is when we would first start to see cord formation based on their developmental stage. But what I was really surprised to find is when we did histology of the gonads, that in fact, they develop testicular cords by day 2 postpartum. You can see just the outline of cords here in the gonad, and they become really distinct by day 3. And you can see here's our SOX9 staining, showing our Sertoli cell differentiation in these gonads just 2 days after birth. Now, this is much, much earlier in development than you see gonadal development in the Tama wallaby. And I was really, really surprised to see this. It was unexpected completely. So when I then looked at what was happening in other marsupials, I started reading any paper that talked about when cords formed in any marsupial species, is that it always seems to be within 2 days of birth, regardless of whether you're one of these tiny little dasyurid babies, intermediate sort of bandicoot or opossum baby, or a more, you know, developed macropodid baby, regardless of the fact that these are much more developed than these guys down here, all of these will develop cords within 2 days. Now, I guess it's really important to determine sex as early as possible because you want to pattern all of those other tissues appropriately. So you want to get that happening as early as you can in the embryo. And so what this showed us is that the development of the gonad is not related to developmental timing in the marsupial, but rather birth. And you cannot determine sex until after birth. So I started thinking with marsupials with this very simple placental structure, maybe the barrier to them determining sex is really that maternal circulating estrogen. They cannot determine a testis while they're connected to that maternal circulation. But once they're born, they can immediately determine a testis, and then that can lead to the patterning of the rest of their tissues. So I thought I had some great epiphany about, you know, how this process worked. And I started again reading all the literature about what other people have done in the past with other species. And I actually found a paper by Ursula Mitwock, who's an amazing geneticist that worked on sex determination a number of years ago. And she had actually proposed exactly this for the monodelphis, where she showed that if she prolonged pregnancy in the monodelphis, she actually delayed cord formation. And in every instance, cord formation was kept on hold until after that animal had given birth, no matter how long she delayed pregnancy for it and birth. And so it was a really fascinating example of, you know, sort of digging back through the literature and seeing that this is something that somebody else had suggested. And we now have a lot of proof of that really being the fundamental mechanism and understanding now why, and that it's estrogen that's probably preventing testicular determination now. And you can see, obviously, the marsupial placenta that you see here is a very different structure to this mouse placenta, which is able to metabolize out all of that estrogen and prevent it from getting across to the baby. So they can determine sex in utero, but marsupials can't. So marsupial mode of reproduction then, it seems like, is probably happening just because they cannot determine sex inside. So they have to have that baby born at a very early developmental stage, chuck it out into mum's pouch, then it can determine sex, and then they can go on with the rest of their development. This is really in contrast to us thinking that mammalian reproductive mode is really an adaptive characteristic that help them regulate their own pregnancy in this really harsh Australian environment, where this kangaroo here, if it's, you know, falls on hard times and can't support the young, it can literally chuck it out of the pouch and mum can survive. Whereas if you're a eutherian and you have a pregnancy and multiple babies, you're really committed to that pregnancy right to the very end of it, even if it's going to kill the mother in harsh times. So I think it's really helping us shed light on the evolution and why marsupials might have this strange reproductive mechanism, I think is really just to get that baby out as soon as possible so you can determine sex, and then the rest of development has to occur then in the pouch. So in addition to all these developmental resources for the Dunnart, we're also doing a bunch of genetic resources, which I won't bore you with, but we're also developing a bunch of cell lines for this animal as well. So we've got iPS cells and spermatogonial stem cells that we're growing for the Dunnart. with the hope that we can do some genetic manipulations to really start to help these guys out. Spermatogonial stem cells are a really great method for working on conservation genetics because we don't need to transfer embryos into assisted reproductive technology to get the benefits of gene editing if we use spermatogonial stem cells. So we're trying to grow these cells, culture them, and then get them to colonize recipient testes so that animals can then pass on genetically modified sperm that may have advantageous characteristics within it. So I see that we're running late on time, so I'm going to skip some of our strategies for what we're doing and just move straight to my conclusions here. But there's a number of different genetically modified marsupials that we're really interested in generating for conservation purposes, and also really finessing our ability to derive these stem cells or iPS cells so we can start to create really good biobanks for marsupials. So one of the things I'm sure you're all aware of is the horrific bushfires that we had in Australia last year. And we lost a lot of fragmented marsupials populations, particularly really diverse and important populations. So we're hoping now that we'll have robust methods for iPS cell derivation. We can go out, collect tissues from those marsupials. We can biobank that diversity and then hopefully develop methods for reintroducing those back into the population at some later time point. So overall conclusions with our lessons from the pouch, I think I hope I've shown you that marsupials are really excellent and unique models for studying reproduction. They've really helped us change the way we think about the evolution of sex determining mechanisms, and particularly the role of estrogen in that process, which has lots of implications for endocrine disruptor impacts to us and wildlife. So really important for conservation efforts and also human health and reproductive health. We're developing new models to help study reproduction in marsupials with the development of the Dunnart, which hopefully we can do manipulation in. And we're also developing those different cell lines and things to really help with marsupial conservation biology to hopefully have, you know, some better mechanisms in there to help prolong these amazing species as diverse as possible. And with that, I'd like to thank the funding bodies that have supported me. I've been very fortunate to get a lot of really great funding from both the US and Australia to support this work, and also all the people on the right there that helped with various aspects of this work. Thank you very much.
Great, thanks very much for that, Andy. Very interesting talk. I'd just like to remind everyone, can you stick around until the end of the webinar so we can launch a bit of a feedback poll at the end of the webinar, and then I'll open it up for questions. Andy, we've got a question from Whipperwee. I'm assuming that the study was performed with ethinyl estradiol, which is a long-lasting estrogen. Have you tried with 17-beta? And also, have you co-treated the pouch youngs with the estrogen receptor inhibitor ICI 182-780 to see if the effect of estrogen?
Yeah, so we haven't used the inhibitor yet. We did the original study was actually with the 17-beta estradiol. And that's because we didn't really need long-lived estrogen when you're doing the daily treatments in the pouch. So we could use the short-lived 17-beta was fine for that. So those initial studies that were done with opossum sex reversal, Tama wallaby sex reversal were all 17-beta. And then when we've done gonadal culture, we normally use EE2, but we've used both. We use the EE2 in culture just because, yeah, it is more stable and lasts a little bit longer. But we've actually shown the same effects when we use diethylstilbestrol, genistein. So we've used a number of different estrogens now in both the mouse, the wallaby model, or the Dunnart model, and also in our human NT2D1 cells. And we see the same effects with any estrogen receptor agonist. But we haven't used the receptor inhibitor yet, no.
We've got a question here from Philippe. Reduction of the anogenital distance is a good indicator of feminization by EDCs. Have you measured the anogenital distance of male wallabies in your atrazine experiment?
Yeah, that's a great question. So the problem with the wallabies is that they don't have anogenital distance because they have a urogenital sinus. So they don't actually have that separation of the anus and the genitals that you see in eutherian mammals. It's right next to each other. So in a wallaby, you'll have them defecating right where the penis is in those early stages of development. So you don't have that separation out. So unfortunately, in the study where we looked in that early time period, there's no AGD to measure. So it would have been, I mean, obviously we do that with the mouse work all the time because it is such a sensitive thing. But for us, we couldn't. And that's why the penis is sort of our next best surrogate, is because it's so hormonally sensitive, particularly for its length. And we could see that was, the thing that was reduced.
On a similar sort of line, is ejaculate collection possible in any of these models, or is it limited to epididymal sperm collection?
You can collect ejaculates from the Tama wallaby. The problem with that is we have to wait 2 years to do that. With the Dunnart, we haven't tried yet. So we're hoping that we can get an ejaculate so that we can do things like motility and things as well. Other than that, we will be able to collect sperm from the epididymis with the Dunnart at least, which will be great to look at as well.
Can you speak to reproductive aging in this taxa in females at all? What's the major issues that are different from Eutherians?
I don't think we know much about reproductive aging in marsupials at all. So they're not, anywhere near as well studied as Eutherian mammals. In terms of the ovarian reserve, I guess you're asking, whether they run out of eggs at some developmental time point. We really don't know. But these are all things, sort of the reproductive aging and things that we hope now we can assess now that we've got the Dunnart as a model. And we've really only started working on the Dunnart for 2 years. And so it's an enormous, it's bad enough working on an exotic species, that's tough enough. But when you go, I'm going to develop a new model, you have to go back and do all the basics, like the developmental time series, when does gonadal development happen, like everything like that, you have to go back to square one again. And so we hope to address all of those questions with this particular model because it is that nice little, mouse lab-based model. It makes all these things a lot simpler. And they have a really short lifespan. They only live for 2 years. So these are the sorts of things that we can really study in great depth, and we have a large number of them. So we will look at those things, but at the moment, yeah, we just don't know.
I think you've done pretty well already in the last 2 years. April Binder is asking, have you identified the binding partner for SOX9 how they bind to the microtubules yet?
So no. So people have shown that SRY and SOX9 just do bind, but I don't know what facilitates that binding. No. And we can see that, as I said before, with that estrogen, you really see a massive stabilization of microtubules within 30 minutes across, again, all of those models. It's this sort of rapid response, and that the SOX9 just becomes then trapped to it. It is only, I think, unphosphorylated SOX9 that binds. So once it becomes phosphorylated, it dissociates from the microtubule and then goes into the nucleus. So there's a number of those parts of the mechanism that we've looked at. And seeing that, how estrogen impacts each of those parts of that particular pathway. And actually, Melanie Stewart has published a number of papers on this just this last year. So they're all just coming out. I think there's 2 that she published just in December, and one will be out shortly that really go into detail about all of those mechanisms and how it modulates MAPK signaling in particular to have that rapid response.
Great. I think we have time for just one more question. Victor Ruthig is asking, since the time to sexual maturity in male Dunnarts is faster than the Tama, have you done follow-up penile length studies in adults to see if penile length can be recovered after various treatments?
So unfortunately, that student graduated and then moved on, which is really annoying. So anyway, we have another student who's on board this year who's going to do exactly that. So we need to treat Dunnarts and now follow it through to adulthood. So we've looked at the gonads, but we haven't gone through to very early stages of development before the phallus was really sexually dimorphic. And now we'll carry it through to much longer, later stages and have a look at all of those parameters in the adult. Because that's what's really important to show, I think, is that we have sort of a long-term health consequence of exposure to some of these chemicals. And then we can go back to macropodid or these kangaroo marsupials and maybe do some more targeted studies in smaller groups that are more manageable to show that this is also happening, not just in Dunnarts or dasyurid marsupials, but also in the macropodid or kangaroo type marsupials as well. But I think once we've got that data from a more amenable model, we can then do sort of more targeted studies in the macropodids that make it a lot more doable and affordable.
Great, thanks very much, Andy. I think we just had the webinar, I think. We've had lots of appreciative comments for you, Andy.
Well, thank you very much. It was a real pleasure. Thank you very much for inviting me to speak today as well. I love talking about marsupials. I could have gone on for another few hours about all the really cool things. So thank you very much.
All right, Andrew, I'm going to go ahead and launch the poll. That way we can get some feedback from the folks. We'll give them about 30 seconds to go ahead and submit their answers for the polling.
Thank you.
Other than that, that about wraps it up. And just a reminder, you can find more about this and other upcoming webinars by visiting www.marsupial ssr.org and clicking on the News and Events tab. And again, thank you, Andrew, for a fantastic presentation.
Thank you.
And thank you everyone for participating. It looks like we have about 92% of the folks that have completed the poll. We just need about one more and we're all set. All right. Thank you, everybody. Enjoy the rest of your day.
Thanks.
Thank you.